4 billion years ago, Earth's atmosphere was a suffocating, reducing soup. Thick clouds of methane and carbon dioxide trapped the heat above violently bubbling volcanic landscapes, leaving the surface practically devoid of free oxygen. Within that hostile chemical mix, the first prebiotic ingredients began to interact. Amino acids and nucleotides combined into complex polymers, eventually forming self-replicating molecules. This early RNA world set the stage for the very first simple, single-celled life to emerge in the oceans. To find the physical traces of those early cells, we have to look to places like the arid, flat-topped mountains of Western Australia. Deep in this red soil, geologists have uncovered fossil evidence dating back 3.5 billion years. Along the ancient, shallow coastlines, specific types of microbes called cyanobacteria grew in dense, layered mats. Over time, trapped sediment and continuous biological growth stacked up to form these dark, rounded, clustered shapes, known as stromatolites. By 2.9 billion years ago, the cyanobacteria living in those layered mats had developed a remarkable metabolic trick. They began using sunlight and water to generate energy, a process called oxygenic photosynthesis. The byproduct of that energy production was molecular oxygen. By splitting water molecules to fuel themselves, these microscopic cyanobacteria introduced a highly reactive gas into an ecosystem that had never experienced it before. That byproduct would eventually reshape the entire planet during the Great Oxidation Event, roughly 2.33 billion years ago. This was the moment free oxygen permanently accumulated in Earth's atmosphere. The sudden atmospheric change was devastating to the existing anaerobic biosphere. Oxygen acted as a poison, triggering a mass extinction of early microbes and reacting with atmospheric methane to strip away Earth's greenhouse insulation, sending massive glaciers creeping across a frozen, barren planetary surface. This timeline shows a glaring chronological gap in the geological record. We know cyanobacteria began producing oxygen 2.9 billion years ago, but the Great Oxidation Event didn't happen until 2.33 billion years ago. That leaves over 500 million years of missing time. If vast mats of cyanobacteria were constantly churning out oxygen for all those millions of years, that highly reactive gas had to go somewhere. It wasn't building up in the atmosphere. Identifying the biological forces behind this slow fuse reveals why the shift to an oxidizing world wasn't a simple chemical trigger, but a protracted struggle between life and its own environment. We know that the oceans themselves acted as a chemical sponge. As oxygen entered the water, it reacted immediately with dissolved iron. Those reactions rained down onto the seafloor, leaving behind distinct, wavy, horizontal layers of rusted sediment, known today as banded iron formations. But the sheer volume of iron and other reactive minerals in the early ocean simply wasn't large enough to absorb 500 million years of continuous oxygen production on its own. Something else was pulling oxygen out of the system. Finding that missing sink is exceptionally difficult because we are looking for biological activity. Soft-bodied microbes and their internal biochemical reactions do not leave physical structures behind in the rock. By comparing DNA to build a molecular clock on a phylogenetic tree, scientists trace modern enzymes backward to a common ancestor. The target is heme copper oxygen reductases, or HCOs, the cellular machinery modern organisms use to safely breathe oxygen and convert it to water. By reading those modern genetic codes and mapping them onto an evolutionary timeline, biologists can accurately reconstruct the behaviors of entire microbial ecosystems that vanished billions of years ago. The genetic data reveals a striking overlap. The DNA tracing shows that the HCO breathing enzyme evolved during the Mesoarchean era, at the exact same time the earliest cyanobacteria started producing oxygen. In the shallow coastal waters, these different species of microbes lived shoulder to shoulder. They were packed tightly together in dense microbial mats. As soon as a cyanobacteria cell split a water molecule and released a tiny bubble of toxic oxygen, a neighboring microbe equipped with that newly evolved HCO enzyme would instantly absorb and metabolize it. Because they lived in such close proximity, this local consumption formed a highly efficient biological sink. The surrounding organisms breathed the oxygen before it ever had a chance to bubble up through the water column. Earth's first breathers were actively scrubbing the oceans of oxygen as fast as it was being made, effectively trapping the planet's atmospheric evolution in a 500 million year holding pattern. That biological deadlock finally broke when cyanobacteria evolved from single cells into interconnected multicellular filaments. This physical transition allowed them to grow larger, scale their metabolism, and vastly increase their geographic distribution. Suddenly, they were producing oxygen much faster than the neighboring microbes could eat it. The biological sink was completely overwhelmed. The excess gas saturated the oceans, bubbled to the surface, and flooded the atmosphere, triggering the Great Oxidation Event. When that toxic oxygen saturated the globe, it wiped out most of the old anaerobic biosphere. But some organisms had already adapted to the gas, including the direct ancestors of all complex life, the early eukaryotes. Microfossils dating back 1.75 billion years show exactly how they survived. These early eukaryotes lived as benthic aerobes, meaning they anchored themselves to the ocean floor and relied entirely on the localized, oxygen-rich patches generated by nearby cyanobacteria. Because the open oceans remained largely hostile and devoid of oxygen, these early eukaryotes were physically restricted to those shallow seafloor zones for over a billion years, waiting for global conditions to shift enough to allow them to expand into the open water. The development of these oxygen-handling enzymes established a new metabolic standard. By refining the pathways to neutralize and utilize oxygen, early microbes provided the high-energy foundation that eventually allowed life to break its size constraints and evolve into the complex multicellular organisms we see today.